Educational. Today we are going to start with a new chapter, the fundamental unit of life from biology, class 9 Cbse. This is your master teacher, SUVOPRIYO, and today we are going to start the chapter. So let's start the chapter without much delay. So if you have ever downloaded a Telegram app from Play Store or iOS, then please join our Telegram channel and all kinds of PDFs, workbooks, notes, positivity, inspiration, motivation will be given in this channel. So join the channel right now. So let's go to the chapter. Yeah. So who was Robert Hooke and what was his discovery or the contribution in the discovery of a cell? So Robert Hooke was the first physicist. He was basically an English physicist and who was successful in observing the cells in the it was in the primary or the earlier microscopes. So let's read what it does. The discovery of the cell was made possible through the invention of the microscope. We know the cell is very much smallest and tiniest part of our body. So it is simply impossible to see the cell by our naked eye. So it was simply possible when the invention of the microscope was done, and the cell was first discovered and named by English physicist Robert Hooke in 1665. So Robert Hooke was basically a physicist, and he was the first physicist who found or observed the cell under the microscope in the year 1665. He made thin slices of cork and likened the body partitions. He observed the cells in the monastery. So the cells were looking the there were partitions between the cells, and they were simply looking like small rooms which were present in the monastery. However, what Hooke actually saw was the dead cell walls of the plant cells, cork as it was appeared under the microscope. So dead cell walls of the plant cells cork. Cork is basically the tree trunk, the hard woody part of the tree. So that part is basically sclerenchyma and they are basically dead in nature, and so for which he was unable to see any kind of it was simply dead, and he was unable to see any kind of organelle or nucleus or other parts of the cell. The cell walls observed by the Hooke gave no indication of the nucleus and other organelles found in most of the living cells. So the cell walls and other parts of the cell were not present in the uh, cork or the structure he was being observing under the microscope. So I think you are able to understand about the discovery of Robert Hooke. So let's move to the next slide. So who was Anton van Leeuwenhoek? Anton van Leeuwenhoek was a scientist from Netherlands. He is known as the first microbiologist because he was the first to observe bacteria underneath a microscope. He was a scientist from Netherlands and he was the first microbiologist. Means the microbiologist means that the scientist who culture with the microbiology. Now microbiology is basically the observation of the microorganisms. So he was the first, and then he was the first to observe the bacteria under the microscope. So he made many other significant discoveries in the field of biology and also made important changes to the microscope. Significantly, he was a scientist who discovered in the field of biology different kinds of observation, different kinds of inventions he has done in the field of biology, as well as he made important, significant changes in the microscope as well. So let's go to the next slide. So discovery of Anton van Leeuwenhoek. Anton van Leeuwenhoek was the first to discover bacteria, smarma, tozoa, erythrocytes, microscopic. crustaceans, single-celled algae, and ciliates. So he was the first to discover bacteria, spermatozoa, erythrocytes, microscopic crustaceans, single-celled algae, and ciliates. So these organisms was first observed and discovered by Anton van Leeuwenhoek. He also established differences in the structure of dicotyledons and monocotyledons as well. He was the first to see the facet structure of insect eyes, lens, fibers, transverse muscle fibers, scales, and tubules of dental substance. He also significantly observed different structures of insects, such as insects' eyes, lens, fibers, transverse muscle fibers, scales, and tubules of their dental substance. So I think you are able to understand the discovery that is being performed by Anton van Leeuwenhoek as well. So let's move to the next slide. So who was Robert Brown and what was his contribution in the discovery of the cell? Robert Brown was a Scottish botanist and paleobotanist. So Scottish, he was from Scotland and he was a botany. He was a scientist from botany and he also used to culture with the means earlier body parts or the anatomy or the structure of the organisms as well, which is a paleobotanist of the plants. So paleobotanist means the structure of the anatomy of the earlier plants. He made important contributions to botany by his pioneering usage of microscope. So he has also contributed different means different inventions he has done by in botany by his usage of the microscope. His discovery of the nucleus and its role helped to prove the cell theory, which states that all living organisms are composed of cells and cells come from pre-existing cells. So the most important invention Robert Brown has done was the discovery of the nucleus, and its role helped to prove the cell theory. He also helped to prove the cell theory as well, which states that all living organisms are composed of cells and cells come from pre-existing cells. So, generation after generation, this nucleus of the cell continues in each and every cell of the living organism. The contribution in discovery and naming of over 2,000 species of plants has been done by Robert Brown. Robert Brown has also discovered and named 2,000 species of plants in this world. So let's move to the next slide. I think the discovery and knowing about Robert Brown is simply crystal clear to you. So let's move to the next slide. Matthias Leiden and Theodor Schwann and their discovery. These both scientists, Matthias Leiden and Theodor Schwann, were from Germany, and one of them is Matthias Leiden was a German botanist, and Theodor Schwann was a German zoologist. So let's read. Matthias Leiden, a German botanist, concluded that all plant tissues are composed of cells. So the plant tissues, now you know that number of cells build up tissues and tissues build up organs, organs build up organ systems. So like that, different plant tissues, all plant tissues are composed of cells. So all the plants in this whole world, in this biosphere, are composed of cells as per conclusion made by Matthias Schleider. Theodor Schwann, German zoologist, he concluded that all animal tissues are composed of cells. So Theodor Schwann concluded that all animals in this universe, all the animal tissues are composed of cells. So he also published that animals' tissues are composed of cells, and so I think the discovery or the contribution of Arthur Schleiden and Theodor Schwann is very much clear to you. So let's move to the next slide. So what is basically prokaryotic cell? Prokaryotic cells are single-celled organisms. known to be the earliest on the earth. So, before the existence of human beings or before the existence of life on earth, prokaryotic cells were the first single-celled or microorganisms which were being found on earth. Prokaryotes include bacteria and archaea. So some of the examples of prokaryotic cell are bacteria and archaea. The photosynthetic prokaryotes included cyanobacteria that perform photosynthesis. So some of the prokaryotes also perform photosynthesis like plants as well. Example like cyanobacteria. A prokaryotic cell consists of single membrane and therefore all the reactions occur within the cytoplasm. So they have a single cell membrane or plasma membrane around their cell, and all the kinds of reactions which is very much useful and necessary for the cell occurs within the cytoplasm. Cytoplasm is basically the liquid structure which is being present inside the cell. They can be free living or parasites. Some of the prokaryotic cell are free living as well as parasite as well. So let's move to the next slide. Yeah, this is the detailed diagram of the prokaryotic cell. So these blue-like globules are simply ribosomes. These red-like structures are plasmid. This green-like structures are pili, and these liquid which is present, this is cytoplasm, and this green structure is a plasma membrane, and over it there is a yellow structure which is cell wall, and totally this is being bounded by a red membrane which is capsule. Now the bacterium flagellum, this thin thread-like structure is a bacteria flagellum, and now nucleoid is this structure, that is the red structure, red thread-like structures. So this is the circular DNA as well, nucleoid. So let's move to the next slide. What are eukaryotic cells? Eukaryotic cells are having nucleus enclosed within the nuclear membrane and form large and complex organisms. So prokaryotic cells were having a nucleoid, but they were not having any kind of nucleus within the cell. But in case of eukaryotic cells, they are having the nucleus which is enclosed within the nuclear membrane, and they are forming large and complex organisms. So most of the multicellular organisms, not most, but all of the multicellular organisms are eukaryotic. Now, examples like protozoa, fungi, plants, and animals all have eukaryotic cells. They are classified under the kingdom Eukaryota. Now, they can maintain different environments in single cell that allows them to carry out various metabolic reactions. They can maintain different environments. Now, different environments are being faced by these eukaryotic cells, and they have the single cell that allows to carry out various metabolic reactions. Metabolic reactions means various energy is being produced by different kinds of reactions, and these are being carried out in these single cells. These helps them grow many times larger than the prokaryotic cells. Eukaryotic cells grows many times larger than the prokaryotic cells as per different kind of environments. So let's move to the next slide. Till that, I think the different type topics we have discussed, I think it is simply crystal clear to you. So let's move to the next slide and discuss about the next topics. So this is a detailed diagram of the eukaryotic cells. So here you can see that this is the nucleolus. Now these, it is being bounded by surface which is having pores over it, and this is the nuclear pore. The nucleolus is present inside a membrane or the plasm, which is the nucleoplasm, and the pore-like structures, it is the nuclear envelope. So after a lot of coverings and membranes, nucleus is being found inside it. So these all are the parts of the nucleus. Now here we can find these structures, these orange structures as rough endoplasmic reticulum. This is the mitochondrion and this blue-like structures, which is the food factory of the cell. Now this liquid is the viscose liquid is a cytoplasm, and these tubules are the secretory vesicles. These are the Golgi apparatus and peroxisome. This is the structure of the peroxisome and centrosome and centriole, lysosome, rhizosomes, and this is the tail-like structure with cilium, cell membrane, which is all the parts present are being bounded by the cell membrane. Now this is a smooth endoplasmic reticulum, and here we are finding the rough endoplasmic reticulum. This is the smooth endoplasmic reticulum, and total detailed structure of the eukaryotic cell. So let's move to the next slide. So, what is the difference between the eukaryotic cell and the prokaryotic cell? So, prokaryotic cell are the relatively simple small cell ranging from 1 to 2 micrometer. Now, complex large cells ranging from 10 to 100 micrometer. Now, these prokaryotic cells are being possess rigid cell wall which prevents rupture of the cell. In the eukaryotic cells, no rigid cell wall is vertebrates, but in plant cell wall is present. Now, membrane-bound organelles are absent in prokaryotic cell. Membrane-bound organelles are present in eukaryotic cell. Genetic material is not enclosed in nuclear membrane. It is contains prokaryotic cell only contains nucleoid. Any kind of nuclear membrane means any kind of genetic material is not present. Only the nucleoid or the nuclear membrane is present. Genetic material is enclosed in the well-defined nuclear membrane. Genetic material consists of single circular or endless double helical molecule of DNA. There are many chromosomes formed of linear open-ended double helical molecules of DNA. So I think this difference between the prokaryotic and eukaryotic cells are very much clear to you. So let's move to the next slide. So what is plant cell? Plant cells are the eukaryotic cells that vary in several fundamental factors from other eukaryotic organisms. Both plant and animal cells contain nucleus along with similar organelles. One of the distinctive aspects of plant cell is the presence of cell wall outside the cell membrane. So plant cells are basically eukaryotic cells. Now that vary in several fundamental factors from other eukaryotic organisms. But both the plant and animal cells contain nucleus along with similar organelles. So this means that plants and animals, the cell structures or the anatomy of the cell, all the structures of the plant cell and the animal cell are not similar to each other. They have difference between them. So, but nucleus and other similar organelles like cytoplasm, then vacuoles, like the plants have large vacuoles, animals have small vacuoles. So these are some exceptions which are being present. But then one of the distinctive aspects of the plant cell is the presence of cell wall outside the cell membrane. Plants needs more protection, so they have another rigid cell wall outside the cell membrane as well. So the plant cell is rectangular and comparatively larger than the animal cell. Plant cell is rectangular in structure and they are comparatively larger than the animal cell in size. Even though Plant and animal cells are eukaryotic and share a few cell organelles. Plant cells are quite distinctive when compared to animal cells as they perform different functions. So the kind of functions played by plant cell are much more in number than animal cell, and they have a more number of cell structures in plant cell than animal cell as well, and so they have different and more functions in animal cell. So some of these differences can be clearly understood when the cells are examined under an electron microscope. So these differences between the plant cell and plant cell and animal cell will be much more clear to us if we examine both of them under an electron microscope. So I think it is very much clear what is plant cell to you people. So let's move to the next slide. So this is the detailed diagram of the plant cell. So here you can see this is very much large in size, and there are some exceptions like you can see that they have uh, mitochondria, lysosomes, cytoplasm. These are very much common. But then you have cell wall, which is this is the exception. Then we have chloroplast. This is the exception. Between chloroplast, we have leucoplast. Then other different parts we have. We will discuss each and every part in this chapter later in this video. So let's move to the next slide. So what is animal cell? Animal cells are range in size from a few macroscopic microns to few millimeters. So animal cells range in size from a few microscopic microns to few millimeters. They are smaller in size than a plant cell. The largest known animal cell is the ostrich egg. Everybody know that in this universe, in this biosphere, among all the egg-laying organisms, the egg cell of the ostrich is the most largest among all of them, which can stretch, and its measurement is it can stretch over 5.1 inches across and weights of about 1.4 kg. This is in stark contrast to neuron in the human body, which is just 100 microns across. So it can be compared or contrasted with the neuron in the human body, which is just 100 microns across. The shape of the animal cells also varies, with some being flat, others oval or rod shaped. Now the shape of the animal cells also varies, with some being flat. Some of the animal cells are flat in nature, others are oval or they have rod shape. And there are some also more intriguing shapes such as the curved, spherical, concave and rectangular. So these shapes of the animal cells may be curved in nature, spherical in nature, concave as well as rectangular. Most of the cells are microscopic in size and can only be seen under the microscope. So animal cells are also very much small, and these cells cannot be seen by the these cells cannot be seen by our naked eye, and so they are being seen under the microscope. So plant cells are also being seen under the microscope, as well as animal cells are also being seen under the microscope. So I think it's very much clear what is animal cell and all its properties. And let's move to the comparison between the plant cell and animal cell. So this is the detailed diagram of the animal cell. So cell membrane is the outermost covering of the cell. Nuclear core, nucleolus, nucleoplasm, nuclear envelope, and Golgi apparatus, secretory vesicle, cytoplasm, mitochondria, cell membrane, cilium, ribosomes, lysosomes, centrioles, centrosome, peroxisome, smooth endoplasmic reticulum, rough endoplasmic reticulum. Each and every part of the cell will be discussed in this chapter. So, is in this video as well. So, yes. So this is the difference between the animal and plant cell. Plant cell having Usually they are larger than animal cells, usually smaller than plant cells. Cell wall present in addition to plasma membrane and consist of middle lamella and a primary and secondary wall. Cell wall is absent. The last or the outermost covering of the animal cell we know as the cell membrane. Plasmodesmata present. Plasmodesmata is absent. Chloroplast present. Chloroplast absent. Vacuole large and permanent. Vacuole small and temporary. Tonoplast present around vacuole. Tonoplast are absent. Centrioles absent except motile cells or lower plant centriole present. Nucleus present along the periphery of the cell. Nucleus at the center of the cell. Lysosomes are rare. Lysosomes present. Storage material in storage trains. Storage material in glycogen granules. So this is the overall difference and all kinds of exceptions or exceptional parts which is present in the structure of the plant cell, which is not present in the animal cells. So this is the basic difference and the comparison between the plant cell and animal cell. I hope you are able to understand about these differences. So let's move to the next slide. So what is plasma membrane or the cell membrane? So the plasma membrane or the cell membrane provides a protection for a cell. Each and every cell needs some protection from their outer outer causing dangers or outer substances coming inside the cell. So they need protection. It also provides a fixed environment inside the cell. It also helps to have a fixed environment or whereby which the cell should be habituated and the cell can survive. And that membrane has several different functions. These cell membranes or the plasma membrane also has several different functions. One is to transport nutrient into the cells and also to transport toxic substances outside of the cell. Different kinds of waste substances are being also produced as well as different kinds of need the substances which are being very much needed by the cell are also being transported inside the cell, and the toxic substances or the waste substances which is being produced inside the cell are being left out by these plasma membrane. Another is that the membrane of the cell, which would be the plasma membrane, will have proteins on it which interact with other cells. So these plasma membrane also have proteins on it which helps to interact with other cells. So this is the basic diagram of the cell membrane or the plasma membrane. So I think you are able to understand the plasma membrane or the cell membrane, as we have said that we will discuss about it in detail and we have discussed it. So I think you are able to understand. So let's move to the next part of the cell. So this is cell wall. So it is an exception in the animal cell. Animal cells doesn't have cell wall. They have the outermost covering as cell membrane, but in plant cells, over the cell membrane, we have another cell wall, which is the outermost part of the plant cell. Cell wall is a structural layer surrounding some types of cells just outside the cell. So this is the outermost part of the cell. It can be tough, flexible, sometimes rigid. It provides cell with both structural support and protection and also acts as filtering mechanism. It is both structural, provides support and protection from the outside danger causing to it. And in algae, fungi and eukaryotes including plants but are absent in animals. So in algae, fungi and eukaryotes, some of the eukaryotes, some are or fungi and algae, all these organisms consist of cell walls except these animals, living animals. All the cell wall is absent in animals. So I think I'm able to understand the detailed discussion about the cell wall. So let's move to the next part of the cell. So nucleus. Nucleus is the important and the most dominated structure of the cell. So let's read. The nucleus is the membrane-bound organelle which contains the genetic material of the eukaryotic organisms. So it is a membrane-bound organelle that contains the genetic material. Now, what is genetic? Genetic means the transfer of characteristics from your parents to the offspring. So this is the genetic. Now, the genetic material or the characteristics which is being transferred in the eukaryotic organisms are being present in this nucleus. As such, it serves to maintain the integrity of the cell by facilitating transcription and replication process. Now, different kinds of facilitation of the transcription and replication processes are being going on in this nucleus. It is the largest organelle. By its structure, by its anatomy of the nucleus, we can see that it is the largest organelle in the whole cell, both in plants, both in animals. So, inside the cell, taking up the tenth of the entire cell volume. So, 10% are being governed by this particular leader or the most important part of the cell, which is known as the nucleus. This makes is one of the easiest organelles to identify under the microscope, as it is so much large in size. So it is very much easier to observe it under the microscope. Some of the other main components of nucleus include phospholipid bilayer membrane. The kind of membrane it is placed inside it is phospholipid bilayer membrane. Then it consists of nucleoplasm. Then it contains the nucleolus, and within this chromatic is present. So this is the detailed anatomy of the nucleus. Hope you are able to understand the important and vital role as well as the structure of the nucleus or in this cell, both in plant cell, both in animal cell. So let's move to the next slide. So cytoplasm. Cytoplasm is the gelatinous liquid or the viscous liquid that fills inside of a cell. So basically, cytoplasm is a kind of liquid or the gelatinous liquid or the viscous liquid which is present in the cell. All kinds of reactions occurs in the cell, occurs in the cytoplasm. So it is composed of water, salts and various organic molecules. Some intracellular organelles such as nucleus and mitochondria are enclosed by membranes that separate them from the cytoplasm. So intracellular organelles like nucleus and mitochondria are enclosed by membranes that separate them from cytoplasm. So these liquids should not enter within these intracellular organelles. That's why nucleus and mitochondria are being covered with a cell membrane so that the viscous liquid or the gelatinous liquid doesn't move inside the nucleus and mitochondria. So this is the detailed structure of the cytoplasm, and hope you're able to understand the structure and the need of cytoplasm inside the cell and what cytoplasm composed of. So let's move to the next part of the cell. So cell organelle. So the cellular components are called the cell organelles. So all the cellular components, all the structure of the cell are known as a cell organelle. These cell organelles include both membrane and non-membrane bound organelles present within the cell and are distinct in structure and function. These cell organelles include both membrane and non-membrane. Some of the structures or some of the parts of the cells like mitochondria and nucleus, they have structures, and some other parts they do not have any kind of structures or the plasma or the membrane. They present within the cell and are distinct in their structures and functions. Each and every organelle within the cells have their own distinct and specific function and structure. They coordinate and function efficiently for the normal functioning of the cell. Each and every cell are working as per their specific function. So the cell can work efficiently and can do normal functioning. A few of them function by providing shape and support, whereas some are involved in locomotion and reproduction. So different cells are being doing their specific function. Some of the functions, like some of the cells are engaged in locomotion of the cell, some of the cells are engaged in the reproduction, and some of the cells are engaged in giving shape to the whole cell. Now, there are various organelles present within the cell and are classified into three categories based on the presence or absence of the cells. So, they are being uh, classified in categories on the presence or absence of the cells. So, hope you have understood the structure and the specific functions done by the cell organelles which are present inside the cell that helps the cell to work normal and to protect the cell from various outer toxic substances as well as outer dangers. So let's move to the next part of the cell. So it is endoplasmic reticulum. It is also very much important structure of the cell. So let's discuss about it. Endoplasmic reticulum in biology, a continuous membrane system that forms a series of flattened sacs within the cytoplasm of the eukaryotic cell, serves multiple functions important, particularly in the synthesis, folding, modification, and transport proteins. In biology, a continuous membrane system, they have a continuous membrane of flattened sacs, and then within the cytoplasm, they are present within the cytoplasm, and they serves different and multiple functions, and being important, particularly in synthesis, folding, and modification and transfer of proteins. They do synthesis, they do folding, they have folding, and then they do modification to the cell, and they do transportation of proteins within each and every part of the cell. Now, all eukaryotic cells contain an endoplasmic reticulum. All the eukaryotic cells, either be it is a plant cell, either be it is an animal cell, each cell consists of an endoplasmic reticulum. In animal cells, the endoplasmic reticulum usually constitutes more than half of the membranous content of the cell. Difference in certain physical and functional characteristics distinguish the two types of endoplasmic reticulum, such as rough endoplasmic reticulum and smooth endoplasmic reticulum. The Golgi apparatus. Now, this is the structure. This is a complex structure. So, Golgi apparatus, also called the Golgi complex or the Golgi body membrane, bound organelle of the eukaryotic cells. Now, which is clearly defined nuclei that is made up of the series of flattened stacked pouches called the cisternae. So, the number of flattened stacked pouches are called the cisternae of the Golgi apparatus. And the Golgi apparatus is responsible for transporting of the modifying and packaging the proteins and lipids in the vesicles for delivery to targeted destinations. So Golgi apparatus are helping the cells for packaging the proteins and lipids to get delivered to different destinations as per they are being specified. It is located in the cytoplasm next to endoplasmic reticulum and near the cell nucleus, while many types of cell contain only one or several Golgi apparatus. Plant cells can contain hundreds. So, many types of cells contain only one or several Golgi apparatus, but then plant cell contains hundreds. So, 100 Golgi apparatus maybe it may be present. So, I think it is very much clear to the about Golgi apparatus of the cell. So, let's move to the next part of the cell, lysosomes. Lysosomes is basically containing digestive enzymes. So the energy which is being produced, these energies are being produced by different kinds of reactions by different kinds of enzymes. So all these enzymes are contained inside the lysosomes. Now lysosome is being bounded by a membrane. So we have discussed previously some of the organelle or the some of the cell organelle contains membrane-like structures. Some of the structures inside the cell doesn't contain membrane-like structures. Among these, lysosomes contain the membrane-bound structure. Now let's discuss about it. A lysosome is a membrane-bound cell organelle that contains digestive enzymes, and lysosomes are involved with various cell processes. Various kinds of cellular processes are being continued by lysosomes, and also they are being involved by lysosomes. They break down excess or worn out cell parts. They may be used to destroy invading viruses and bacteria. It also helps as an immune sustain for the cell to destroy the invaded virus and bacteria. If the cell is damaged beyond repair, lysosomes can help it self-destruct in the process called the programmed cell death. If the cell is damaged or it cannot repair much, if totally or partially the cell is getting damaging by some outer foreign bacteria or the viruses. Now, the lysosomes, the capability of the lysosomes to protect the cell from these foreign bodies, if the capability crosses, means the foreign bodies are too much stronger than lysosomes, then for which the lysosomes bursts out and or breaks out, for which the total cell gets dead or damaged by the lysosomes or cell death or apoptosis. So I hope the lysosome part of the cell is very much interesting, and it is very much able to understand by you people. And let's move to the next part of the cell. So mitochondria. Mitochondria we know as the food factory of the cell. So all kinds of energy produced by the produced and found inside the cell that makes a cell to be survive or to be living inside a body is done by mitochondria. Mitochondria is present both in animal cell, both in plant cell. The energy currency produced by the mitochondria is ATP, so it is adenosine triphosphate. So before discussing much, let's read what it tells. Mitochondria are membrane-bound cell organelles that generate most of the chemical energy needed to power the cell. Biochemical reactions and chemical energy produced by the mitochondria is stored in a small molecule called adenosine triphosphate (ATP) molecules. Now, mitochondria contain their own small chromosomes. They contain small chromosomes within the mitochondria, and they generally, these mitochondria and therefore mitochondrial DNA are inherited only from the mother. So, different kind of inherited characteristics we have inside the cell, but then the mitochondrial DNA is being inherited only from the mother. So, let's move to the next part of the cell, but then before it, I think mitochondria is very much crystal clear to you people. So, what are plastids? Plastids is an exception to the plant cell. These plastids are not present in the animal cell. Plastids are the substances which give color to the particular plant. For example, if the plant like tomato is in red in color, so the plastids present in the tomatoes are in red pigment. They are having red pigment. For different kinds of spinach like spinach, cauliflower, or cabbage, the plastids present in these uh, plants are green in color. That's why we can see that they are green in color. So before discussing much, let's read what it is. Plastids are double membrane organelles which are found in cells of plants and animals. They are not present in animals. So plastids are responsible for the manufacturing and storing of food. They are simply responsible for the manufacturing and storing of the food. These often contain pigments that are used in photosynthesis and different type of pigments that can change the color of the cell. They contain pigments, different pigments, and that are used in photosynthesis. So the plants which are non-green. So the plants which are non-green, those plants are having different plastids. So they can they are used for photosynthesis. So those plants have different pigments and they can change color of the cells. So these plastids will not be every time would be green in color like normal plants. I hope plastids are very much clear as they are the exceptional part towards the plants and not present in animals. So I think it is very much clear and it is very easy as they are being the exceptions. So let's move to the next part. So this is the detailed structure of the plastids. This is the outer membrane, this is the intermembrane space, and this is the inner membrane, and this is the stroma, aqueous fluid. This is the granum, this is the thichaloid, and this is the lamella and lumen. These green-like structures are known as the granum, the stack of thichaloids, they are known as the thichaloids, and stack of thichaloids are called the granum. And inside the thichaloid, each inside each thichaloid, these thichaloids consists of lumen, and these stacks of thichaloids are being connected to each stacks of thichaloid by lamella. So types of plastids. So one type of plastid is known as chloroplasts. Chloroplasts are biconvex shaped, semi-porous, double-membrane cell organelle found within the mesophyll of the plant cell. They are sites of synthesizing the food by the process of photosynthesis. So these are biconvex shaped and semi-porous. These are double membrane. Double layer is being present over the cell for its protection and for it giving the shape, and cell organelle found within the mesophyll of the plant cell. Within these cell organelle, the mesophyll cell, plant cell is being found, and they are sites of synthesizing food by the process of photosynthesis. So the process of photosynthesis we know, by which plants can process to make their food by taking water, by taking carbon dioxide at the daytime, and by taking the sunlight or the solar energy, and all these mixes up, and the whole procedure is being continued in these chloroplasts. So the structure is intermembrane space, and this is a stroma lamella, by which each and every thichaloid or the stacks of thichaloid are being connected, and inside the thichaloid they are being consist of lumen, inner membrane, and this is the inner membrane in which the stacks of thichaloids are being present, and this is the outermost membrane, and these stacks of thichaloids are known as the granum, and this is inside this there is a viscose or the liquid present in it is the stroma. So let's move to the next type of plastid. So it is the leucoplast. Leucoplasts are a category of plastid, and as such are organelles found in the plant cells. They are non-pigmented and contrast to other plastids such as the chloroplast. So they are the category of the plastid, and as such are organelles found in the plant cells. These are found only in plant cells, as plastids, as we have discussed previously itself, plastids is only present in the plant cell, and among these plastids there are three parts. One of them is leucoplast. So leucoplast is also present in the plant cell, and they are non-pigmented. It doesn't contain any kind of pigments. So without consisting of any kind of pigment, they are not having any kind of color. So in contrast to other plastids such as the chloroplast, Lacking of the photosynthetic pigments, chloroplasts are not green. So we have discussed now only that without having any kind of pigment, they are not green in color and are located on non-photosynthetic tissues of plants such as roots, bulbs, and seeds. So we have never found that roots are green in color. So leucoplasts are present in roots. We have never found that bulbs are green in color. So leucoplasts are present in bulbs. And as well as seeds are not green in color ever. So leucoplasts are present in seeds. So this is the microscopic view of leucoplast in this image, and I think the topic leucoplast is very much clear to you people. So let's move to the next slide. So another type of plastid is chromoplast. Chromoplast are the plastids. It is heterogeneous organelle responsible for pigment synthesis and storage in specific photosynthetic eukaryotes. So these are plastids, heterogeneous organelles responsible for pigment synthesis and storage in specific photosynthetic eukaryotes. So photosynthetic eukaryotes contains of chromoplast. Now it is thought that like all the other plastids, including chloroplast and leucoplast, they are descended from symbiotic prokaryotes. So it is thought that like all other plastids, including chloroplast and leucoplast, they are descended from symbiotic prokaryotes. So symbiotic, they are descended in symbiotic prokaryotes. And then chromoplasts are found in fruits, flowers, roots, and streaks and edging leaves and responsible for their distinctive colors. So different kinds of colors are being found in chromoplasts, and this is always associated with massive increase in accumulation of carotenoid pigments. So these are always associated with massive increase in accumulating of carotenoid pigments. And the conversion of the chloroplast to chromoplast is ripening is a classic example. So conversion of chloroplast to chromoplast in ripening is simply a classic example. Hope you have understood the type topic chromoplast, which is one of the types, and it is the last type of the plastid. So let's move to the next slide. So it is the vacuoles. Now vacuoles are present both in plant cells and present both in animal cells. So the term the animal cell contains of small vacuoles, and then the plant cell consists of large vacuoles. Now the term vacuole means empty space. They help in the storage and disposal of various substances. They helps in the storing of various substances. They also helps in the disposal of various substances. They can store food or other nutrients required by the cell to survive. They also store waste products and prevent entire cell from contamination. The vacuoles in plant cells are larger than those in the animal cells. The plant vacuoles occupy more than 80% of the volume of the cell, and the vacuoles may be one or more in number. So the vacuoles in plant cells are larger than those in animal cells. We have discussed this beforehand. And the plant vacuoles occupy more than 80% of the volume of the cell. So vacuole. So the organelle called vacuole we can see in the plant cells, and when plant vacuoles occupy more than 80%. 80% of the total volume of the cell can found plant vacuoles. So plant vacuoles may be one or more in number. Not necessarily that vacuoles will be in one number. It may be that vacuoles will be more in number in the cell. So this is the structure of the vacuole, which is being mentioned in this image. Hope you have understood vacuoles. What is the structure of the vacuole and how much vacuole is covering the area of a plant cell. So cell division. Cell division happens when a parent cell divides into two or more cells called daughter cells. So cell division occurs in two or more cells. It may happen that cell division occurs in more than two parts. For the daughter cells, cell division usually occurs as part of the large cell cycle as cells reproduce by splitting into, while each parental cell gives rise to two daughter cells. So splitting of a parent cell into two cells is called the daughter cell. there are two distinct types of cell division, out of which the first one is vegetative division, wherein each daughter cell duplicates the parent cell called mitosis, and the second one is meiosis, which divides into four haploid daughter cells. Now, there are two distinctive types of cell division. One is vegetative division, in which the daughter cell is simply duplicating the parent cell. Means the parent cell is being divided into two, and these cells are simply duplicating, and this process is known as mitosis. The second one, or the second distinctive type, is the meiosis, in which the parent cell is being divided into four haploid daughter cells, and they are not simply duplicating with the parent cell. So next, let's move to the next slide. So this is the mitosis and this is the meiosis. So prophase, this is the prophase part, this is the prometaphase, this is the metaphase, anaphase, and this is the telophase. So this is the formation, and after this telophase we can see two daughter cells. Now in this prophase one we can see that metaphase, anaphase, and in this we can find telophase, and after that we can see it is forming two parts, and then we can see it is making a formation of four parts. This is the four haploid daughter cells. So let's move to the next slide. So this is the end of this chapter. So thank you, and hope all your concepts are simply crystal clear from this chapter. And if you have any kind of doubts, mention it in the comment section below. And till then, we make a lot of efforts for making such conceptual videos. So like the video and then subscribe the channel and hit the bell icon to get regular updates, and then share the video as much as you can. So till then, stay tuned.